Alright, so we're going to pick up now with section 5 on the acoelomates and pseudocoelomates, and we're moving into the tapeworms. So remember now, we're still in the phylum Platyhelminthes, we're in the subphylum Neodermata, and we're in the class Circummeromorpha. The old name for the tapeworms is Cestoda, which I had on a previous slide back in section 4. Some people still call them cestodes, but Circummeromorpha is a more frequently used name now for this group. So same organism, but different name. Now with the tapeworms, and again, I can't stress this enough. This is the most advanced group of the flatworms, and I say that because they have the most specialized traits that they use for making a living and for being a parasite. So one of the things you see right off the bat in this picture is they're going to have a series of hooks up here on the front, which they can use to embed themselves within the host's typically intestine. They're going to have suckers, which they can also use to hold on to their host tissues wherever they happen to occur, usually in the intestines, but not always. They don't have eye spots because it's dark in there. And another feature they have that we haven't talked about yet is a lot of times these tapeworms will have a covering over their body which sort of shields them from the immune system of their host. So it's a way to kind of like blend in with the host tissues so the host's immune cells don't try to fight these off and destroy them. So another sort of characteristic feature of the tapeworms. They, like we've said before, do not have a GI tract. They're going to absorb their nutrients right through their cuticle, right through their skin, essentially. So a very thin outer covering to absorb nutrients, which have, of course, already been digested by a host. This front segment here is called the scolex. And the scolex, like we talked about before, is the part that has hooks and suckers and so forth. This is essentially the head of the tapeworm, even though it doesn't really look like a head of anything, really. That is the frontmost segment of the tapeworm. Behind this scolex is the neck, which is unsegmented. And we get just a little bit of a sense for what that looks like down here. This is a scanning electron micrograph, by the way, of this organism. This is the neck down here. And if you were to move posteriorly behind the neck, you would actually also run into what are called proglottids. And so most of the body of the tapeworm is going to consist of proglottids. These are the individual functional units of the tapeworm that do the bulk of the work for the tapeworm. And we'll talk about really what those consist of here in a few minutes. But essentially, as you move from the front of the animal toward the back of the animal, the proglottids are going to get bigger and bigger and bigger, and also more and more mature. And eventually, proglottids are going to break off from the posterior part of the animal, and those proglottids are then released with the feces of the host into the environment, where they then distribute eggs into the environment as well. So we'll talk about exactly how that works here in a few minutes. Most of the tapeworms, or a lot of the tapeworms, fall into the genus of Taenia. In the case of humans, there is a single human tapeworm called Taenia saginata, which has humans as the primary host, right? So the host where reproduction takes place. And in the case of Taenia saginata, the secondary host is actually cows. So cows are the other host, which helps the tapeworm get back into humans. So we talked about the scolex, right? We've got the neck, which is the first part, which is unsegmented. And then behind that, every single block here is essentially a single functional proglottid, which is it's basically an egg factory is what it comes down to. Each proglottid is going to be able to produce hundreds of thousands of eggs, which are then fractured off of the tapeworm and moved into the environment with the feces of its host. So these proglottids are, like it says, hermaphroditic. They are monoecious. They are able to self-fertilize. So, they still get some of the benefit of sexual reproduction when it comes to genetic diversity, but they don't get the full benefit because they're not reproducing with a partner. So it's kind of like a compromise between, not having to find a host, but still getting some of the diversity that comes along with doing meiosis and producing gametes. So the tapeworm can actually live for a year or two inside of a host. They can have a fairly long lifespan, and they continuously make new proglottids, like an assembly line for proglottids, which eventually mature, self-fertilize, and then break off and leave the host and move into the environment. All right, so we'll take a look at the life cycle of Taenia saginata here momentarily. This tapeworm here, scolex, neck is somewhere up here. These are fairly small proglottids, but as we move toward the back, you can see how big these proglottids actually get by the time they're mature, and by the time they start breaking off into the environment. These are big enough that if you happen to be infested with a tapeworm, you would very much know it. You would essentially, I guess if you pay attention when you go to the bathroom, try to be delicate here, you would notice those functional proglottid units leaving the body as you went to the bathroom, we'll say it that way. So, the bigger ones are definitely visible with the naked eye. You get a sense for how big. This is not a human tapeworm here, by the way. This is some other species, but you get a sense. Okay, so here's a foot, 12 inches here. So you imagine, this entire length of this thing might be 20 or 25 feet long. That's a pretty substantial mass of tapeworm there. The scolex is somewhere up here. It's very small front end. This is where it was holding on, and the proglottids would be getting bigger and bigger and bigger as you move toward the posterior most part of the animal. And then we've got just a light micrograph here of a gravid or full of eggs proglottid as well. You get a sense for the scale here, 3 millimeters, so definitely visible, barely with the naked eye, but certainly not very large. All right, so let's take a look at the life cycle here of the tapeworm. So just kind of for consistency, we're going to start off the same way we did with the flukes. We're going to start off with the proglottids and the eggs within, leaving the body of the human host with the feces. And so these proglottids then exit the body, they end up in the environment, and so they're out there now on the ground, essentially, where you might potentially have cattle grazing. Right? So this is, tapeworms, again, are not a very common thing in the U.S. Typically when people get tapeworms in the U.S., it's because they've traveled outside the U.S. and they've picked them up somewhere else. But it can happen here, but the chances of it are very, very, very slim. So if you have human waste in the environment, and you have cows grazing in that same part of the environment, then you have essentially what you need for tapeworm infestation to occur. These proglottids can contain, a couple 100,000 eggs. They can be on the ground, in the soil. They can be in the vegetation. They can be in the water. And so if a cow is grazing and picks up some of those eggs from the proglottid, the larvae of the tapeworm are going to form a mass inside the tissue of the cow, the muscle of the cow, essentially, the beef. And so if a human comes along and consumes that piece of beef with the parasite in it, and again, this has to be uncooked or undercooked. If it's cooked thoroughly, this tapeworm cannot survive. But if it's uncooked or undercooked, the tapeworm parasite can survive that, can make its way into the human host, and can set up shop in the intestine. This represents the stomach. So it's after the stomach and the intestine here of the human host. And at that point, it begins to grow. It feeds on nutrients that the human is digesting, and eventually starts releasing its own proglottids into the environment. Now, you might wonder, okay, what's the effect of this on the body? How does this hurt you? Does it hurt you? How bad is it, right? Most people, if they were to have one or two tapeworms, aside from noticing it when they went to the bathroom, maybe being freaked out by it, probably would not be too affected by it. Because a tapeworm, yeah, it's gonna steal some of your nutrients that you're digesting with the food that you eat, but it's not going to be enough to really cause any sort of like starvation or loss of weight or anything like that. Now, if you had a couple dozen of these things inside your intestine, well, now you get the sense that you might actually start running into the possibility of a blockage. And if you have a blocked GI tract, that can actually be fatal, right? So if you get a blockage to the point where it actually causes a rupture of the intestine, that in most cases is not going to be something that somebody survives because of the sepsis that would occur as a result of essentially fecal matter and bacteria being released into the body cavity with other tissues and organs. Very, very dangerous situation and typically lethal if that were to occur. All right. Now, another phylum that I want to look at, and this is one that we're only going to look at for like a slide or two. This is the Acoelomorpha. And the reason why I like to talk about this one is this is another really good example of a case where a group of animals was reclassified when new information was discovered about them. So the Acoelomorphs used to be considered a subgroup of the flatworms. So they were classified right in there with the Turbellaria, the Trematoda, and the Circummeromorpha as somewhat like a side group of the Platyhelminthes. But on further inspection, what they noticed was that they actually do have a nervous system. They do not have a digestive cavity of any sort. And so it turns out that those two traits, right, lack of nervous system and lack of digestive tract, are not because they share those traits with the flatworms. It's because they came up with those traits on their own independently. So this is a case of convergent evolution. And so convergent evolution, if you remember, is a situation where two organisms or two groups of organisms eventually evolve the same traits, not because they're related, but because they live in similar environments and they're faced with similar challenges. And so therefore, it's not too surprising then that they come up with the same set of adaptations to cope with those challenges in their environment. So the Acoelomorphs, if you look at them genetically, on molecular evidence sorts of things, are actually fairly different from the flatworms, even though superficially they resemble them at a morphological level. Like visually, they look like them, even though they're not really that closely related to them. So they're not, for our purposes in Principles 2, they're not so important for their own sake, but they are important because it illustrates, okay, yeah, these species are not hard and fast, set in stone. They can be moved around. Groups can be changed if new evidence comes along, and B, the whole idea of convergent evolution. So that's the other kind of important reason when we talk about those. Another phylum I like to talk about, this one again, not so important for its own sake, but it shows you just how small a phylum can be and how specialized a phylum can be. The phylum Cycliophora was discovered only back in 1995, so it's not really all that old as far as being known to science. Very, very small phylum, very few species. They look superficially kind of like a rotifer, which we haven't covered yet, but we're going to see that here coming up shortly. But they're very, very specific in that they live only on the mouth parts of a certain kind of a lobster in the North Atlantic Ocean, both the North Atlantic, Canada and northern U.S., as well as North Atlantic over in Europe. Cycliophora are very specific to certain kinds of lobsters in that part of the world. So not a very big phylum at all. It's not like you're looking at mollusks where you've got maybe 60 or 70,000 species, or you've got arthropods with over 1 million species. This one you've got like a handful of species for the entire phylum. So another kind of unique offshoot for that reason. Okay, so from there, we're going to move into the pseudocoelomates. We're going to put the acoelomates behind us, those that don't have a body cavity. We're done talking about those. And we're going to talk about a couple of groups of pseudocoelomates, the nematodes and also the rotifers, which if you look down here on this kind of family tree of the animals, you see that they're really not all that close on this family tree. So when we talk about the rotifers and nematodes together in this lecture, again, it's not a situation where they're especially closely related because they're really not, but they happen to share a fairly important common trait, that being the presence of a false body cavity or a pseudocoel. So, again, convergent evolution, they both came up with the same trait because they face similar environmental challenges, not because they're related. So to remind you, a pseudocoel is a situation where you have the gut in the center of the gastrula of the animal, juvenile stage or embryonic stage. You then have an endoderm. You then have a separation, a space, a pseudocoel, a false body cavity, before you move into the mesoderm. And then from the mesoderm, you move directly into the ectoderm. So you've got a complete separation between endoderm and mesoderm in these pseudocoelomates. That's what defines them as a body form. Now, in this case, the pseudocoel is fluid-filled, and that's going to perform the function of a hydrostatic skeleton, like we talked about back in our previous section, a couple sections ago. Essentially, the fluid in the cavity can be pushed against by muscles, and as you compress the fluid pushes back, and that allows the muscles to actually function and move a part of the body. So this skeleton is not based on any sort of hard structure, but it's based on a fluid-filled cavity, and that being the pseudocoel in this group. And again, like I said, this is not a monophyletic group. The two groups that we're talking about, the rotifers and nematodes, are similar only in the sense they both have a pseudocoel. Other than that, especially at a molecular level, they're actually very, very different from each other. All right, so we've got our false body cavity plan here on this picture. We talked about the hydrostatic skeleton. We talked about how muscles work against it. Pseudocoelomates do not have, again, a circulatory system. They're not going to have a respiratory system. They're not going to have an excretory system. They're still fairly simple, but having a pseudocoel does allow them to become larger and more complex in some very important ways. So we're going to talk about those ways here in a few minutes. Our first phylum here, and this is by far the bigger of the two phyla that we're going to talk about, is the phylum Nematoda. Now, if you look at the sheer number of species of nematodes, there aren't as many species of nematodes known to science as there are, say, mollusks or arthropods especially, or even vertebrates. But nematodes haven't been studied as well because they're hard to find, they're small, they're microscopic, they're less charismatic, right? They're not as cute or cuddly as vertebrates are, for example. So, it might be that there aren't as many species out there. That might be why there haven't been as many described, but more likely, the reason why there aren't as many described species is because they haven't been studied as much, and so therefore not as many species have been discovered. And we're going to see a couple of groups like that as we move along. Groups that are probably bigger in reality than what they appear to be on paper. All right, so getting back to roundworms. Most people call them roundworms. Some of the older names refer to their eel-like movement as they swim or as they crawl. Vinegar eels, eelworms, they move in this fashion, this sort of undulating side-to-side fashion, because they only have one layer of muscle. Now, when we talked about the flatworms and when we talk about the segmented worms, they're going to have two layers of muscle. They're going to have both circular and longitudinal muscle, whereas the nematodes only have longitudinal muscle. They have muscle that runs parallel to the long axis of the body this way, but they don't have muscle that wraps around the body this way. So that's going to give them, when they contract the muscle, the body's going to move side to side. It's going to kind of like slither back and forth, almost like an eel swims in the water, which is how they get their eel-like designation. Now, I mentioned that there aren't that many species of nematodes known to science, but if you look at the sheer number of individual animals, nematodes are hands down the largest of all. All right, so if you were to count the number of individual animals, the number of individuals of nematodes, there are more of them than any other group of animals. Okay, they're very, very cosmopolitan. They're very small, which means they can divide up the niches very, very finely and therefore result in the production of a lot of species. There are tons and tons of individuals of each species. So much so that it's been said, and I think I have this on a slide coming up, that if you were to remove all traces of human civilization from the planet, you would still see the outlines of buildings and cars and people walking around because nematodes cover all of those surfaces as well. Right? They're very much everywhere. You could dig up, literally, if you were to take a shovelful of soil from the right kind of environment, you could find 1 million nematodes in that shovelful of soil. That's 1 million animals in what amounts to basically a pound or two of soil. So they're very small, but there's a bunch of them, right? And they can live in any habitat. So saltwater, freshwater, they can be parasitic, they can be free living. For those that are free living, they can be herbivorous in different ways. They can be predators, they can be detritivores, kind of like the flatworms were that we saw, the free living ones. So lots and lots of habitats as well. All right, very, very big group when it comes to number of individuals and the jobs they perform. Only about 20,000 species, but like I said, most people estimate there are at least hundreds of thousands of species, if not more, just based on how many are discovered every year. There's a whole lot out there waiting to be described still. We talked about the abundance a second ago, so these two kind of like visuals I kind of already gave you, I guess ahead of the slide. Got ahead of myself a little bit, but yeah, they're super common. You find them everywhere. Guaranteed you have had nematodes on your body or still do at some point during your life, and for the most part, they don't really cause you any harm. They're what we describe as being commensalistic. Commensalist is a symbiosis where one species benefits from the other species, and the second species is neither harmed nor helped by the first one. So if we sort of put this in this terms, if you have species A and B, if A benefits B, but B doesn't really affect A, that's a commensalism. As opposed to a mutualism where A and B both help each other, or a, exploitative relationship where A is benefited by B, and B somehow, or A has a negative effect on B, and B helps A, that kind of thing, like a plus-minus association. That would be more of an exploitation. Competition is where both species hurt each other. Commensalism is one where if you have something living on your skin that doesn't hurt you, but you're providing it a habitat, you're providing it food, you're providing it a place to live and safety and those kinds of things, that would be a commensalism. And we have bacteria like that on our skin. We at times have nematodes like that on our skin as well. They don't really affect us in any way, but we actually provide their habitat. So that's an example of a commensalism. Now, the nematodes are the first of two groups of ecdysozoans, and we referred to these back in, I think, our very first chapter that we covered. Ecdysis is a process where an organism sheds its outer covering, essentially its outermost layer of skin. If they do that, they are going through what's called ecdysis. Nematodes have an outer layer called a cuticle, which is a non-living layer that gives them essentially waterproofing, and in some cases allows them to evade their host immune system, kind of like we saw with the tapeworms. Nematodes do this, arthropods do something similar as well, and so for that reason, both nematodes and arthropods are classified as ecdysozoans. Now, the reason why they need to shed their outer covering is because that outer covering is non-living, and as such, it doesn't grow, it can't expand as the animal grows. So if the animal wants to get bigger, it has to get rid of its outer covering and grow a new, bigger outer covering to accommodate its now larger body. So think about a snake shedding its skin. Now, in that case, the snake isn't technically doing ecdysis. It's a different process, but it looks superficially kind of similar, where the snake gets rid of the old skin, grows some new scales, grows in the process. Nematodes and arthropods do something similar, but the details of it are somewhat different, I guess you could say. So that's kind of what they're doing here. We talked about their longitudinal muscles as well. They don't have any circular musculature, so that gives them their eel-like movements, and they're going to do gas exchange through their cuticle. As far as distribution of nutrients and waste within the body and gases as well, that's going to be through simple diffusion because they don't have any systems to accomplish any of those tasks. In terms of sensory structures, they do have quite a bit more actually than most other animals we've looked at so far. They can have eye spots. They can have chemosensory structures that they can use to smell or detect various chemicals in the environment. They do have also what are called stylets. So stylets are a type of piercing, stabbing mouth part, which they use to either kill their prey or perhaps feed on part of their prey. Maybe if they're a plant-feeding nematode, they can inject their stylet into the plant and feed on sap, something like that. Stylets are also something that mosquitoes have. That's what they use for feeding. They jab their stylets into your skin and take some of your blood, kind of the same idea. So nematodes have a mouth part like that they use to accomplish their feeding. Whether they're predatory, whether they're herbivorous, whether they're detritivorous, they use their stylets for feeding. And they also have a pharynx like we saw before as well. Now, in terms of reproduction, the roundworms are dioecious for the most part. They are gonochoric, which means there are separate male and female individuals. They are usually not going to be sexually dimorphic, so typically it's really hard to tell a male versus a female roundworm, although there are exceptions, as we're going to see. One of the ones that infests humans is a really good example of a sexually dimorphic dioecious animal, Ascaris lumbricoides, which is a very large roundworm, about as big as they get actually, close to it. That one actually is sexually dimorphic, as we'll see. They also undergo direct development, meaning that, we talked about this before as well, the juvenile stages look very much like the adult stages, the only difference being their size. So that's also something that we can look at as well. We talked about niches. There are parasitic nematodes for just about every plant or animal species that's ever been examined. This last bullet point on there about the placenta of the sperm whale, you don't need to know about that. It was sort of like a fun fact. A 9-meter long roundworm is pretty freaking huge. It's 30 feet or so. That's a big roundworm, but most of them aren't going to be that big. In fact, most of them are microscopic or borderline visible with the naked eye. So some are a little bit bigger. Like I said, Ascaris lumbricoides is a pretty good size for humans, but a lot of them are quite a bit smaller. There's about 50 or so nematodes that are parasites on humans. We're going to take a look at maybe a half a dozen of them and talk about some life cycles and some kind of unique and gross stuff about them, but we're by no means going to go into all the different types of roundworms that parasitize humans. Something else that's unique about nematodes is their development pattern, and they are extremely consistent developmentally, where every single individual, as it attains adulthood, will eventually arrive at the exact same number of cells. So they are eutellus, in other words. And this actually has made them a very, very good candidate for study in the developmental biology world, for people who look at genetics of animals, for people that look at how animals develop through their life cycle. Having a species that's very, very specific. So Cinerabidus elegans, for example, when it's an adult, has exactly 959 cells. Precisely. No exception. Not 958, not 960, 959. So they're very, very consistent, which makes them a really good candidate for study. You take a lot of the variability out of the equation, it makes it much easier to kind of arrive at the underlying processes by which organisms develop. So they've been used a lot for genetics and developmental biology for that reason. All right, so in this slide, we're going to start going into some of the nematodes that affect humans in some way, or at least humans and some of our man's best friend sorts of organisms. And then from there, we'll move into the last stages of this chapter.